Welcome to Cell Clips. In this video, we will be discussing intermediate filaments, one of the three main types of cytoskeletal elements. The cytoskeleton serves many different functions in the cell, such as providing cell structure, forming adhesions between cells, and helping the cell move and divide. There are three unique types of cytoskeletal filaments: actin, also called microfilaments, intermediate filaments, and microtubules. Of these three types of filaments, microfilaments are the thinnest, microtubules are the thickest, and as their name suggests, intermediate filaments are in between the other two, with an average diameter of 10 nanometers. The primary function of intermediate filaments is to provide structural reinforcement of the cell membrane where it contacts other cells or the extracellular matrix. Intermediate filaments are polymers composed of alpha helical monomers assembled together. Just as a rope is made of many smaller strings, intermediate filaments are made from many thin polymers joined together. First, two monomers coil together to form a dimer with an asymmetric head and tail. Two dimers then stack together in an anti-parallel and staggered conformation, making them symmetric. Many tetramers bind to each other to form the mature intermediate filament. Although it doesn't form this way, intermediate filaments are composed of long strands called protofilaments. Two protofilaments associate to form a protofibril, and four protofibrils are joined together to form an intermediate filament. The result is a rope-like filament that is much more flexible than actin or microtubules. These filaments extend between cell junctions to hold cells together. There are several different types of intermediate filaments, each specific to certain tissues and cell types. Intermediate filaments can be categorized into 6 groups with unique properties and functions. Type 1 and Type 2 intermediate filaments are made of keratin. Within this group, Type 1 filaments protect epithelial cells, while Type 2 filaments are found in hair, fingernails, and scales. Among Type 3 intermediate filaments, the most abundant is vimentin. Vimentin filaments strengthen cell membranes and anchor organelles in place in the cytoplasm. Vimentin is the most universally expressed intermediate filament across cell types. Some Type 4 intermediate filaments come together to form neurofilaments and axons. Neurofilaments strengthen the axon and control the speed of nerve impulses by regulating axon diameter. Type 5 proteins, also called lamins, are found exclusively in the nucleus, making up the nuclear lamina. The lamina forms a rigid mesh just beneath the inner membrane of the nuclear envelope. This mesh serves many functions, such as strengthening the membrane and regulating DNA replication. Finally, nestin, one of the type 6 intermediate filaments, is found primarily in nerve cells and is thought to regulate axon diameter as well as the assembly of other intermediate filaments. So, to recap, intermediate filaments come in many different forms due to the variety of intermediate filament monomers. Different types of filaments have unique expression levels in specific cell types. These alpha helical monomers assemble into dimers and then staggered tetramers. Many tetramers bind together to form a mature filament. These filaments are flexible and rope-like, providing structural reinforcement to the cell, as well as connections between cells. Well, that's it for this video. Thanks for watching Cell Clips.